So hello, my name is Dave Morgan. I'm from the University of California in San Francisco. And in this lecture, I'm going to go over some of my own work on studies of how the cyclin-dependent kinases drive the events of the eukaryotic cell division cycle. Now, it's well established at this point that the major regulators of the eukaryotic cell cycle are the cyclin-dependent kinases, or CDKs. And the basic idea is that a series of CDK cyclin complexes are activated in a specific sequence during the cell cycle to trigger the events of the cell cycle in the appropriate order. And so, for example, S-phase CDK cyclin complexes form in late mitosis or in late G1 and are then activated at the beginning of S-phase to initiate DNA synthesis. And then M-phase CDK cyclin complexes form at the end of G2 and are activated to initiate the events of mitosis and take the cell to metaphase. So the big question that I want to address today is how is it that these CDKs actually drive these cell cycle events? Now, obviously, CDKs are protein kinases, which means that the most likely mechanism by which they promote cell cycle events is through the phosphorylation of other proteins, which then bring about those events. And so over the past 10 or 12 years or so, we've dedicated quite a lot of effort to identifying the substrates of the cyclin-dependent kinases in the hope that that will lead us to an answer to this question of how the CDKs actually initiate cell cycle events. And so in this lecture, I'm going to tell you about two methods that we've used to systematically and comprehensively identify CDK substrates. And then in the second half of the lecture, we'll go into some interesting ways in which we've used those lists of substrates to address some general questions of CDK function and phosphoregulation. So the first method we used to identify CDK substrates began about 10 or 12 years ago in a collaboration with Keyvan Shokat, a chemist here at UCSF. Now, Keyvan came up with an idea whereby it would be possible to label the specific targets of a protein kinase in a crude cell mixture. And this slide attempts to explain that basic method. On the left, let's focus on the left at first. On the left is a wild-type regular protein kinase like CDK1 with its cyclin regulatory partner. And typically, when one wants to label the targets of a protein kinase like CDK1, you simply provide that kinase with a version of ATP in which the gamma phosphate is labeled with a radioactive tag. And then when that protein kinase uses that ATP, it will then transfer that P32 onto its substrates. Now, unfortunately, this cannot be used to identify unknown substrates of CDKs, because if you take a pure CDK and some gamma-labeled P32 ATP and put that into a crude cell lysate, you will get not only the labeling of the CDK's targets, but the labeling of all the other protein kinase targets in that lysate, because that ATP can be used by any kinase. And so Keyvan Shokat's idea was to avoid this problem by using so-called analog-sensitive protein kinases. And the strategy is based on the fact that protein kinases tend to contain a large hydrophobic residue in the wall of the adenine binding pocket of their active site. And so the basic strategy is to mutate that large hydrophobic side chain there to a glycine residue, resulting in the formation of an extra pocket in the side of that ATP binding site. And as a result, this mutant kinase can now use a bulky ATP analog in which extra moieties have been added to the adenine base of the ATP. And so, for example, N6-benzyl ATP can be used by the analog-sensitive CDK1 kinase, but cannot be used by a wild-type kinase because that bulky ATP analog can't fit into the wild-type active site. And so, of course, if you put a radio label on the gamma phosphate of this bulky ATP analog and then add this kinase to a crude cell lysate, what you hope to get is the specific labeling of just the direct targets of that protein kinase and no other kinases in the cell lysate, because those other kinases can't use this bulky ATP analog. So this method, we developed this method in collaboration with Keyvan Shokat a number of years ago and applied it to the yeast CDK1 kinase. And the results with that are shown in the next slide. So this slide shows an autoradiograph of a protein gel in which we've separated the reaction products from 3 different reactions, 2 of which are control reactions and the third of which is the experimental reaction. In the first lane, what you see is what happens when you add this radio-labeled N6-benzyl ATP, this bulky ATP, to a crude cell extract made from yeast. And the result is that you get very little labeling of anything in that cell extract because that ATP analog cannot be used by the protein kinases in that crude cell extract. The next lane is another control in which we're mixing the purified protein kinase, CDK1 AS1, together with a cyclin partner, and then adding that to some N6-benzyl ATP that's radiolabeled on its gamma position. And the result then is that you see autophosphorylation of the cyclin subunit of the CDK cyclin complex. And so that results in a background band in the experimental lane over here. But the third lane is really the crucial lane in which all three components have been added. And so we're adding a purified kinase, analog-sensitive kinase, with the bulky ATP analog and the cell extract. And the result is that you see a whole raft of different proteins being radiolabeled in this cell extract. And those proteins are presumably the direct targets of CDK1 cyclin complexes in that lysate. So we obtained this result a number of years ago and then dedicated quite a lot of effort to identifying these various radiolabeled bands in this cell lysate. And to make a very long story short, we ended up using proteomic libraries to individually identify substrates, and in the end we came up with a list of about 181 proteins in cell extracts that are rapidly modified by CDK1 cyclin complexes. And so this list of proteins provided us with our initial list of CDK substrates. These substrates are involved in a wide range of different cellular processes, many of which are known to be connected to the cell cycle in some way, and are likely to represent important targets of CDK1 throughout the cell cycle. But for various reasons, we decided that this list of substrate was incomplete. And also, because it was done in vitro, we wanted to get another approach that would allow us to identify comprehensively a larger number of CDK substrates that were modified in vivo by CDK1. And so the second method we've been using more recently has been to use quantitative mass spectrometry approaches to identify all the phosphorylation sites in the cell that are dependent on CDK1. In other words, phosphorylation sites whose levels decrease abruptly when you inhibit the protein kinase activity of CDK1. And that method begins again with the analog-sensitive CDK1 mutant. Now, another advantage of these analog-sensitive mutants is that not only do they bind bulky ATP analogs, but they also bind bulky inhibitors that can only fit into the active site of the analog-sensitive kinase, but not the active site of a wild-type kinase. And so, for example, this inhibitor here, 1NMPP1, binds with extremely high affinity to analog-sensitive CDK1, but has essentially no affinity for the wild-type kinase or for any other kinase in the yeast cell. And so we could use this analog-sensitive CDK1 to make a yeast strain in which we can inhibit CDK1 in vivo rapidly and specifically. And so we did that a number of years ago. We created a yeast strain in which the endogenous CDK1 protein is replaced with the analog-sensitive protein. And in that yeast strain, it is now possible to almost completely and specifically inhibit CDK1 activity within minutes by the addition of 1NMPP1 to the culture medium. So we used that strain in this quantitative mass spectrometry approach that I want to tell you about, which was done in a collaboration with Judith Vilaine and Stephen Gygi at Harvard University. And the basic approach that we used is illustrated in this slide and in the next two slides as well. It begins, as I said, with the analog-sensitive yeast strain, CDK1-AS cells, in which CDK1 can be inhibited specifically with the 1NMPP1 inhibitor. And what we do is we grow two parallel cultures of this yeast strain. One culture, the so-called light culture, is grown in regular lysine and arginine, whereas the so-called heavy culture is grown in a different form of lysine and arginine, in which carbon-13 and nitrogen-15 have replaced the usual carbon-12 and nitrogen-14. And so as a result, after growth in this medium for some time, all the proteins in these cells have been labeled with slightly heavier than average lysine and arginine residues, which means that all the peptides derived from this culture will have a slightly higher mass in the eventual mass spectrometry analysis. And that will allow us to identify peptides coming from these two lysates. So we treat the heavy culture with the inhibitor, 1NMPP1, for a brief period, 15 minutes, and then we harvest these cells after the inhibitor treatment, harvest the cells, mix them together, lyse them, break them open, and then treat all the resulting proteins in those cell lysates with trypsin to break them all down into tryptic peptides. And then Judith Vilaine in the Gygi lab has developed a wide range of powerful methods for purifying the phosphopeptides out of that tryptic peptide mixture, and then we then subject those phosphopeptides to mass spectrometry, as shown in the next slide. There are two basic forms of mass spectrometry that are applied to these phosphopeptide mixtures. The first, on top, is to use conventional tandem mass spectrometry methods to actually fragment these peptides and use those fragments to determine their sequence. And so by this approach, we can determine the sequence of all the phosphopeptides coming out of these yeast lysates, and just as importantly, we can identify the precise site of the phosphorylation on those peptides. And so by doing this, Judith was able to produce a list of about 10,000 phosphorylation sites on 2,000 different proteins in the yeast lysate. And then in addition to determining sequence, we also quantify all the peptides and determine the relative amount of the so-called light and heavy peptides. What this means is that every peptide coming out of these phospholipeptide mixtures comes in both a light form, which originally came from the light medium culture, and a heavy form that originally came from the inhibitor-treated heavy culture. And they can be distinguished based on this slight mass difference of their lysines and arginines. And what we're looking for, of course, are peptides that look like this, where the heavy peptide is much less abundant than the light peptide, and that means that that peptide's abundance was inhibited or decreased as a result of CDK1 inhibition, and therefore that phosphorylation site on that peptide represents a CDK1 dependent phosphorylation site in vivo. And so by applying this approach to the many phosphorylation sites identified here, we came up with a list of about 547 phosphorylation sites on about 308 proteins that were clearly CDK1 dependent and represent likely candidates for CDK1 targets in vivo. This list of targets included many of the same proteins we had identified in our previous screen in vitro, and so for those proteins at least, we have very good evidence that these proteins are kinase substrates both in vitro and in vivo. Now the list of substrates includes a wide range of proteins involved in a wide range of processes. I'm not expecting you to see or read any of the gene names on these lists here. This slide is simply meant to illustrate that we have lists of proteins involved in a wide range of interesting processes. Some of these processes are totally expected. For example, DNA replication, spindle behavior, kinetochores, and cytokinesis are all processes in which we expect CDKs to be involved in regulating some aspect of those processes. There's also a few surprises here as well. Protein translation, chromatin structure, and nuclear transport, and the secretory pathway all have a number of CDK substrates involved in those processes, and so one might imagine that this will lead to some new understanding of how CDKs might control those processes as well as the more conventional cell cycle regulated processes. But for the rest of this lecture today, I'm not going to talk in detail about any specific substrates or processes, but instead I'm going to tell you how we used our list of substrates to address some interesting general questions in how cell cycle progression is controlled by CDKs in general. And so we're going to address two questions in the remainder of this lecture, the first one of which is shown on this next slide. And that question is this one: how do different cyclins trigger different cell cycle events? So I told you at the beginning of the lecture that S-phase cyclin CDK complexes initiate S-phase, and mitotic CDK cyclin complexes initiate M-phase. And there is good evidence from yeast genetics and elsewhere that S-phase CDK cyclin complexes have a better intrinsic ability to initiate S-phase than a mitotic cyclin CDK complex. So there's something different about cyclin CDK complexes that are activated at S-phase that allows them to more effectively activate the onset of S-phase. And so what is that difference? Well, one obvious possibility is that the cyclin that associates with a CDK helps determine the substrate specificity of that CDK. So in budding yeast, for example, where there's only a single CDK associating with all these different cyclins, one can imagine that associating with an S-phase cyclin at the beginning of S-phase might target that CDK for specific substrates involved in S-phase. And so we decided we could address this question on a more global level by actually analyzing the relative phosphorylation rate of a wide range of different CDK substrates using purified S-phase CDKs and M-phase CDKs. And specifically, we carried out these studies using the S-phase cyclin CLIB5 from budding yeast and the M-phase cyclin CLIB2 from budding yeast. And Mart Logue, a postdoc in the lab, basically purified these two kinases and then tested their activity towards about 150 different CDK substrates to look for substrates that were highly specific for one or the other. Some of his early results are shown in this next slide, which gives you an illustration of the sort of thing we found. Here we're looking at autoradiographs of protein gels in which three different proteins listed across the top, MCM3, ORC2, and ORC6, have been treated with either the mitotic cyclin CDK complex on the left or the S-phase complex on the right. And you can see quite clearly that these three proteins are all phosphorylated much more rapidly by the S-phase CDK cyclin complex CLIB5. So Mart went ahead and did this exact same reaction with about 150 proteins, as I said, and the results from those experiments are shown on this slide. So this slide summarizes everything that he found. What we're looking at here is a plot of about 150 proteins, each one of which is represented by these little circles on this plot. And these circles are plotted according to the rate of their phosphorylation by CLIB2 on that axis and CLIB5 on this axis. And so most of the proteins are falling along the diagonal of this plot, indicating that they are equally well phosphorylated by both kinases. In other words, they're not cyclin-specific targets. However, we found a quite large number of proteins over here on the right, especially these red circles here, that represent proteins that are far more rapidly phosphorylated by CLIB5 CDK1 than they are by CLIB2 CDK1. So these proteins, and note by the way that this is a log phase scale here, so some of these proteins are 10 or over 100 or even 1,000-fold more rapidly phosphorylated by CLIB5 CDK1 than by CLIB2 CDK1. So these clearly represent proteins that are highly CLIB5 specific, that the cyclin is somehow determining or increasing the rate of phosphorylation of these proteins. So what are these proteins? Well, we were satisfied to see that at least 5 of them are proteins known to be involved in DNA replication, especially SLID2 here. SLD2 is a protein whose phosphorylation is known to be crucial for the initiation of DNA replication. And so these proteins make perfect sense as CLIB5 specific targets because those are the proteins that we need to phosphorylate early in S phase to help drive progression through chromosome duplication. So with this list of cyclin-specific substrates in hand, we next addressed the mechanism underlying this cyclin specificity. Why is it that CLIB5 CDK1 phosphorylates these proteins so much more rapidly than CLIB2 CDK1? Through kinetic studies, we discovered that the reason for this higher rate of phosphorylation was that these substrates have a much higher affinity for the CDK cyclin complex when CLIB5 is associated, suggesting that they might associate with that cyclin subunit. In fact, there's previous suggestions of what the mechanism for this association might be. And those are based on the known crystal structures of CDK cyclin complexes from human cells. So this shows the crystal structure of a CDK cyclin complex from humans, that illustrates very nicely the basic parts of the CDK cyclin complex and where cyclin substrates typically associate with this complex. Now, over on the left is the CDK catalytic subunit, and between these two lobes here is an active site cleft in which you can see this ATP molecule binding right here. And typically a protein substrate would bind along the surface of this protein kinase right here in a way that the serine or threonine hydroxyl would be positioned in such a way to allow the transfer of phosphate from that ATP onto the hydroxyl residue. So the primary site of substrate association with a CDK cyclin complex is, of course, the active site, the place where that serine or threonine associates with its sequence context to be phosphorylated. However, this is probably not the only site of substrate association in a CDK cyclin complex. There's considerable evidence from mammals and from yeast as well that there's a docking site on the cyclin itself that can also associate to some extent with parts of a substrate. And this docking site is mostly composed of this large alpha helix here that contains a number of hydrophobic residues that are together called the hydrophobic patch that is involved in associating with certain substrates and enhancing activity towards those substrates. So we obviously hypothesized that perhaps this docking site on CLIB5 exists on CLIB5 and that this docking site is required for the cyclin-specific phosphorylation that we saw in our experiments. And so to test that, the obvious approach was to mutagenize this docking site through a number of single point mutations and then test whether that has any impact on cyclin specificity. And that is shown in this slide. And the answer was a definite yes, that mutation of that docking site completely abolishes the CLIB5 specificity that we had seen. So here again, we're looking at autoradiographs of protein phosphorylation by purified CLIB2 on the left 2 lanes and CLIB5 on the right. And we're looking at the phosphorylation of 5 highly CLIB5 specific proteins. And you can see that the wild-type CLIB2, the WT here, phosphorylates these proteins rather poorly, whereas wild-type CLIB5 phosphorylates them extremely well. Once again, indicating how specific these proteins are for CLIB5. However, if you mutate the hydrophobic patch or the docking site on CLIB5, you find that that specific phosphorylation is almost completely lost, indicating that that site is really required for the increased affinity that CLIB5 CDK1 has for these substrates. So we conclude that an interaction, probably simultaneous between this docking site and the active site, allows specific CLIB5 substrates to interact with the CLIB5 CDK complex in a high affinity fashion that allows more rapid phosphorylation of those proteins. And so that leads us to at least a partial answer for the question that I first posed, which is, How do different cyclins drive different cell cycle events? Well, part of the answer appears to be that the associated cyclin that associates with a CDK helps target that CDK to specific substrates. And so S-phase CDK cyclin complexes, when they're activated at the end of G1, tend to phosphorylate more rapidly the proteins that are most important for initiating S-phase. Okay, now I want to turn to an entirely different sort of general question that we also used our substrate lists to address. And in particular, we used our recent mass spectrometry analysis and our 547 CDK1 dependent phosphorylation sites to address this question. And this is a much more general question than just that goes beyond issues of simple cell cycle control, but reaches into areas involved in the general issues of phosphorregulation. And the question is this one: how is it that phosphorylation changes the function of a protein? How is it that the addition of a phosphate group to a protein changes that protein's function in a way that allows it to initiate cell cycle events or do other things? And there are typically a couple of different approaches or different mechanisms that are thought to be involved in changing protein function. And the first and possibly most commonly imagined mechanism is this one here, the so-called allosteric switch. And the idea with this mechanism is that the placement of a phosphate on a protein in a very specific location causes a precise conformational change in that protein that then initiates some change in its function, its enzymatic activity, or its association with something. Now, this mechanism, of course, requires that the position of that phosphorylation site is extremely precise and conserved. In other words, you can't put a phosphate just anywhere on a protein and cause this very precise conformational change. It has to be extremely well positioned, and because of that, it's very difficult to evolve that sort of phosphorregulation. That kind of phosphorylation site cannot appear randomly very easily and achieve the kind of regulation that is required. So the alternative mechanism is this one, which I call bulk electrostatics. And this mechanism suggests that the position of the phosphorylation does not require such precise positioning of the phosphorylation. The basic idea here is that the placement of clusters of phosphorylation sites on the surface of a protein, typically on a loop or a disordered region on the surface, can result in interesting regulations, such as interference with association with another protein, or for that matter, promotion of association with phosphate binding proteins. And so this very simple mechanism of phosphorregulation can occur by the placement of clusters of phosphates in a general region of a protein, but the exact position of each of those phosphates is not absolutely important, not critical, and therefore the position of those phosphates can shift during evolution in different proteins. And so for that reason, this mechanism is much more easily evolved. It's very easy to imagine that random mutations could result in the appearance of phosphorylation sites on the surface of certain proteins where they interact with other proteins, and that can result in regulatory possibilities that could be selected for. And so both of these mechanisms are known to be important in different cases. There are examples of proteins that are regulated in both of these ways, but we thought that perhaps our giant list of CDK substrates would allow us to address the relative importance of these two mechanisms more generally. And so what we did was we took those 547 CDK1 dependent phosphorylation sites and aligned them with homologous sequences from other species to see how well these phosphorylation sites are actually conserved. And the basic idea here was that if we found that sites are generally extremely well preserved, that might argue for this sort of mechanism, but sites that drift during evolution might argue for this sort of mechanism. So the next slide gives you an illustration of the sort of thing that we found. Now here, we're aligning a bunch of different protein sequences, and I don't expect you to actually read these sequences. The important thing is that there are these little yellow boxes that represent SP or TP dipeptide motifs that are the consensus sequence for CDK phosphorylation. And along the very top here is the sequence of a part of a protein called SHP1 that we identified 2 phosphorylation sites in our mass spectrometry experiments. Those sites are site A and site B. One site is over here in a region of the protein that is known to form into a globular domain, and another site is here in a region that's predicted to form a disordered domain. And these other sequences that lie below this top sequence from budding yeast are the sequences of orthologous proteins from various yeast species whose genomes have been sequenced, starting with the most closely related yeast here and moving all the way down to the most distant related yeast at the bottom. And so these protein alignments tell us some very simple things. First of all, site A here is very well conserved in evolution. And you can see that almost all of the orthologs of this protein and all these other yeast species contain a likely CDK consensus site at the exact same position in this highly conserved region. So site A appears to represent an example of the kind of site I mentioned in the left side of the previous slide, a site that is highly conserved in evolution. But site B is not. Site B is very poorly conserved and disappears essentially after a few species and is no longer found at that position in other orthologs. However, if you look in this region of these other species proteins, you find that SP and TP dipeptide motifs appear scattered throughout this region in a large number of these yeast homologous proteins, suggesting that even though the initial position here in budding yeast has not been preserved, the CDK phosphorylation of this region has been conserved in evolution, but the exact position of the phosphorylation sites has been shifting dramatically over evolution. So this is obviously consistent with the second idea, that precise phosphorylation site positioning is not required here because these sites might be involved in some more simple general regulatory mechanism involving association with phosphate binding proteins or interference with protein binding. So we did this exact same alignment for all 547 of our phosphorylation sites. And then in the next slide, what I'm going to show you is a somewhat complex graphic that illustrates the results that we found from that. And this was done in a collaboration with Brian Tooch, a graduate student working in the laboratory of Sandy Johnson at UCSF. And this top plot here represents a hierarchically clustered clustergram, as we call it, that illustrates the conservation of the precise position of phosphorylation sites in orthologs of the proteins we identified. And so what we're looking at here is a graphic in which there are 547 columns in this graphic, each one of which represents a single CDK1 dependent phosphorylation site that we identified by mass spectrometry. And then each row in this graphic represents how that phosphorylation site aligns with its orthologs in other species on the same yeast species that I showed in the previous slide, starting with the most closely related yeast species and then working to the most distantly related ones. And in each column, a yellow box indicates that phosphorylation site is precisely conserved in its position in that orthologous sequence. In other words, over here on the left, this yellow box at the top moves down for a few species and then disappears, indicating that this site is only precise, that these columns here, these phosphorylation sites, are conserved only in the closely related yeast species, but then are lost in all distantly related species. And so by looking at this graph, you can see that there's only a small group of phosphorylation sites, these ones in here especially, and particularly these ones here, that are conserved throughout all the yeast homologs that we identified. And so this small number of phosphorylation sites, perhaps 30 or 40 of them, are preserved in large numbers of yeast species, indicating that the precise position of phosphorylation has been conserved in a relatively small number of cases. Okay, so how do we then test the possibility that that instead of precise positioning of evolution, during evolution, we're looking at drifting phosphorylation site positioning. And that required the development of another graphic, which is shown below here, and which I'll take you through slowly because it's a little bit complicated. So in this case, once again, it's another hierarchically clustered graphic in which there are 547 columns, each representing a different phosphorylation site identified in our analyses. But in this case, and once again, the rows represent alignments with orthologous proteins from other yeast species. But in this case, the yellow box doesn't indicate precise positioning of phosphorylation site, but instead indicates that the ortholog of that particular protein in this other yeast species has a statistically enriched frequency of CDK consensus sites, SP and TP motifs. In other words, these yellow boxes represent proteins in which the frequency of SP and TP motifs is far greater than that expected by chance. And so these large numbers of proteins here represent proteins in which, even though the precise site of phosphorylation is not conserved, as shown up here, these proteins do contain a high frequency of CDK consensus sites whose positions are clearly drifting during evolution. And so these large numbers of proteins over here on the right side of this clustergram may represent proteins in which the precise position of phosphorylation does not matter, but that the regulation of those proteins by phosphorylation is conserved despite that. And so clearly, we'd like to think that evidence tends to suggest that this mechanism on the right here, this easily evolved bulk electrostatic mechanism, is a major mechanism by which phosphoregulation can easily be evolved, and that drifting phosphorylation sites, especially clusters of phosphorylation sites on disordered regions, is really crucial for the regulation of many different CDK substrates. So with that, I want to leave us with the question that we started out this whole lecture with, and that is, how do CDKs drive cell cycle events? Well, clearly our lists of CDK substrates, the ones I'm showing here and the many that aren't shown here, probably contain the answer to this question. Clearly through the detailed analysis of large numbers of these substrates, and that will lead us to a much better understanding of how CDKs drive the events of cell cycle and how they alter all these different processes in the cell to initiate cell cycle events.